canine, one of the canine tapeworms, is the dog's the definitive host, but the intermediate host is ruminants. If a ruminant gets inside there, or gets in the cycle, then the dog has to eat the ruminant, or eat parts of the ruminant that has these cysts inside that ruminant, okay? So in this case, proglotid is passed, proglotid disintegrates, and eggs are released. Then the egg hatches, and it gets into the intermediate host. Once it's in the intermediate host, then it's going to get into the portal vessels of the ruminant. And once it gets into the portal vessels, then it can go anywhere it wants to go. Okay, migrate into the different organs of the viscera. Then it matures. Once it matures into a cysticercus, now dog comes along and either eats a dead one or kills the ruminant like a sheep, and then eats the organ meat. And then they get the cysticercus inside them. It then matures in their intestine, and it starts to attach to the small intestine. This it has an armed scolex. That means the proboscis area has got spikes on it. And that's what sticks into the intestine. We can see this by looking at a segment under a microscope. They look very distinct. They look like a teeny tapeworm. They've only got a pore on one side. And then all that you really see is the reproductive organs. Or look at the egg. The egg has a typical tinea appearance. When you look at that, it has kind of a thick shell. A one cell stage in there, but then you can see the hexacant inside of it. The next is tinea ovis. It's pretty much the same thing as an indirect life cycle. That means it needs to have that intermediate host. And in this case, everything is essentially the same thing, but now we just have a different species. It looks different. The egg looks a little different, but there's still a hexacant in there. But the life cycle is still essentially the same thing, but in this case, instead of getting into the organ meat, it gets into the muscles. And that's where this one insists, and the cysticercus is then eaten by the dog. Now we get into Echinococcus granulosus, and this is a really bad tapeworm. It's also one of the smallest tapeworms. The dog is the definitive host in this case. The intermediate, because again it's an indirect life cycle, is going to be a herbivore. A sheep is one of the more common ones. But unfortunately, people can become an intermediate host, or essentially a dead-end host, unless the dog eats a person. Then it can go complete a cycle in the dog. This one has the proglotid that's passed out. Then it's ingested by the intermediate host. In this case, it would be hopefully a sheep and not a human. Once it hatches, then you get this larval form that helps to, or forms a hydatid cyst in the organ meat. And a hydatid cyst is this massive fluid-filled thing that has these little baby tapeworms, if you will, inside of it. This hydatid cyst can be massive, okay? Each one of those little unilocular protostolaces becomes an adult. And then once the definitive host eats it, then it attaches to the intestine. Do you notice where this is coming from? You see that? That's a human surgery. It's coming out of the brain. Hydatid cysts can be anywhere, and they cause serious, serious problems. They can be inside your organs, they can be in the brain. One egg, if you eat that, it can then get into you. So if you get one of those Echinococcus eggs and it gets in your mouth, then that hydatid can go anywhere it wants to go. This is such a bad disease that it's reportable to the government. You have to report this to the state, and they will report it to the CDC. The definitive host for Echinococcus multilocularis, again, is going to be either a cat or a fox. The intermediate host in this one is a rodent, but humans can be in there too. All we have to do is ingest the ova. So the proglottid is passed out, it's ingested by the intermediate, whether it be a rodent or a human. In this case, that hydatid thing forms in the organs, and it's a multilocular, not just unilocular. There's little pockets all over the place. And inside of that, there are these scolexes or protoscolices, and each one of those is going to be a new tapeworm if it gets eaten by the definitive host. These things are very invasive, and they will go anywhere inside. So here's a rodent. Every one of those tiny little white things you see in there, that's a scolex. And you can see how it's got little pockets all over the place. Those can form inside people, okay? Echinococcus is really a bad thing. This doesn't have that big fluid-filled bag that you saw in the unilocular hydatid cyst, but it has this multilocular, all over the place pockets that fill up with those things. Again, this one is passed to people if they ingest the egg. Obviously, they got to get this taken care of. They got to go to the doctor, but this is also reported. If we diagnose this in a patient, we've got to report it to the state, and they will then report it to the CDC. And then they're going to come and try to figure out where it's coming from. Now, these hydatid tapeworms, when you look at these things, you can see the eggs on the fecal flotation. So whether it's Echinococcus multilocularis or Echinococcus granulosus, you see these tapeworms, eggs, and they look very similar. You can't really tell them apart. So all you really know is they look like a taeniid egg. In order to identify this tapeworm definitively, you have to see the adult. And the only way you're going to see the adult is you've got to get them out of the GI tract. And so one of the things that's done for these is they give the dogs that might be infected, they give them a gastric purging agent called orecalin. And then they catch all the feces and then they start looking for these guys. Okay? And that's pretty much it. And if you remember, in the first lecture, I showed you a tapeworm next to a paper clip. And the paper clip was massive, and the tapeworm was just tiny. That's this one. Okay? How small they are. You can do a ELISA test, a blood test, to find out if they've got it. If they've got the antigen-antibody type reactions on the inside there. The big thing for this is to prevent it, because once they get it, it's a big, big concern. So there's actual vaccinations for sheep. And then if a dog gets it, you start treating them. You never feed a dog what? Raw sheep, raw guts from a sheep. You never let them eat that stuff, especially if you're out in the country. All right, the next one is a tetratheridium tapeworm. This one primarily is Cestodes. This can get in dogs, it can get in cats or carnivores. This tapeworm has two intermediate hosts. One of them is called an orbatid type mite. Then the next one is a mouse or a reptile. Obviously, you've got to have the proglottid passed. The egg is ingested by the first thing, and that's that orbatid mite. Then along comes, it develops inside there, the orbatid mite into a cysticercus. Then along comes a rodent, ingests the first intermediate host, and inside of that, then a tetratheridium develops. And these are solid body, but they have exponential reproduction. It just continues and continues. So here's what happened to that mouse sedated. He's completely filled now with all of these solid body tetratheridium things. When you look at it, the egg doesn't look like a taeniid tapeworm at all. it has almost kind of a smooth, corrugated type or folded shell, but you can still see the hexacanth on the inside. When these things grow and they're big, the Scolex does not have hooks in it. It's just got suckers that suck onto the intestinal wall. The next tapeworms are the pseudo-tapeworms or Cotyledus. The one primarily to talk about here is Spirometra. In this case, the dog or the cat can be the definitive host. Because it has an indirect life cycle, there's going to be intermediate hosts. And in this case, there's two of them. The thing about these tapeworms is that the intermediate hosts are generally aquatic species, like a little aquatic crustacean, like that little copepod there. These things are really small crustacean type things, okay? That copepod then is going to allow it to develop into a procercoid. Then along comes a fish or a reptile or an amphibian of some sort in the water and eats the copepod. Once it now gets into the fish or the aquatic animal, then it's going to continue its development into a pleurocercoid. Once it gets to be a pleurocercoid, now something's going to eat the fish or the reptile or the amphibian. And it's going to then to be able to develop inside the definitive host. These things do not look like typical tapeworms, and their eggs don't look like a typical tapeworm. It almost looks like a fluke egg. Okay? There's a little trap door on the end of it called an operculum. But it's the life cycle that makes it different. And then when you look at these things, if you look at that top picture, the Scolex area isn't really there. It's almost like some slits along the side of it that attach. That's why they call these pseudo-tapeworms, because they don't have that kind of Scolex. But this is potentially zoonotic. Okay, and the zoonotic potential is if we eat the copepod or swallow the copepod, maybe you take a drink out of a little creek and there's little copepods that you can't see, but you swallow them. Now that's one way you can get infected. Or you eat the undercooked fish that has the pleurocercoid in it. If you do that, then you get this thing called spiraginosis, where they get into all sorts of tissues. And they just leave it there. And they stay there. They can't complete their life cycle, but that larval stage can be in your tissues for greater than 20 years. And that's what a picture of a human where they made an incision over that larval thing and they're trying to get it to come out now. Okay? And they can get pretty big. Diphyllobothrium latum is another one that can get into dogs and cats and people. Again, two intermediate hosts, aquatic crustaceans again, and then fish. In this case, we have an unembryonated egg that is passed out. That is going to hatch in water, and it creates something called a chorocidium. And that chorocidium starts swimming around in the water. As it's swimming around in the water, this little copepod crustacean comes along and eats it. Okay? Then it develops again to a procercoid. Then it's going to be eaten by a fish, that little copepod thing. And inside the fish, it's going to develop into a pleurocercoid. And then the definitive host eats the fish. To diagnose these, it has the same kind of an operculated egg, looks more like a fluke egg than a tapeworm egg. And you can see the operculum right there. Okay? All right, any questions about up to tapeworms?